Oh, and if I saw the schedule correctly, I'm going to write this and not say it. All right. We talked about this, right? Acid base moving towards a high pH is alkalosis. Moving towards a low pH is acidosis. And a range is 7.35 to 7.45. You are required to know those numbers, 7.35 to 7.45, because they will come up over and over and over and over again. Just to remind you, basic chemistry, the pH scale, negative log of H plus. So what that means is the higher the hydrogen concentration, the lower the pH. Okay, and this is, and then the opposite is true. The lower the hydrogen ion concentration, the higher the pH. So this is just a reminder of those definitions. I know you've all had chemistry, right? And you absolutely loved every second of it.
Chemistry? Yes.
Some people do.
I like open.
Actually, if I were to go back and do things, it would either be physics or chemistry. Do what? Nothing? Who said that? Okay. Let's not talk about physics. All right. It's important to remember, because this is how acid-base issues are set up. You can get an acidosis, in other words, a decrease in pH, by adding more acids to your system or losing bases, right? Because how do you neutralize an acid? You add a base, right? So if you lose bases, you lose the ability to neutralize acids. So that's going to increase hydrogen ion concentration, pH drops, gives you an acidosis. And actually, sometimes you'll see another term, acidemia. That means just acidic blood, and the condition is called an acidosis. Likewise, two ways to become alkalosis. Either you have more bases or you lose the neutralizing factor. You lose the acids. Okay? So keep that in mind as we're going through. You'll see a lot of times the reference range pH 7.4. That's right between 7.35 and 7.45. And what's your death for acidosis? 6.8. What's your death from alkalosis? 8. What do you think is more common? Acidosis.
Does it contain 6.8, or is it right below 6.8? Is considered death?
Once you get into around 6.8, it's not good. One thing you have to realize in physiology and also in medicine, you have to be really comfortable in this area right here. Gray. Or this area. Or this area. All right, the more comfortable you are in the gray, the better off you're going to be, because nothing is ever black and white. That's why a lot of times you guys ask me questions, and I'm like, well, it kind of depends. But there's a difference. If somebody says it depends and never follows up with it, and you're like, really, does it depend? There's reasons why it depends. Okay, so always look for those reasons. It's a neat looking cup. All right, so we've already talked about this, so this is just a summary of what we've already covered. Strengths of acids and bases. You guys know from chemistry that there are strong acids and weak acids, strong bases and weak bases. What our body puts into our body, usually strong acids. What we want to do is convert them to weak acids. Okay, so they don't have as big of a change in pH. So strong acids result in large changes in pH, weak acids, small changes in pH. So our buffer systems in our body, their goal is to minimize changes in pH because we don't want wide swings. We want tiny incremental changes that we can do something about. So our body does a really good job of regulating very tightly with buffer systems, chemical buffer systems, and physiological buffer systems. Okay? Here are your chemical buffer systems. The most important and the most well-known, bicarbonate. That's going to be the most important buffer system for everything. Your blood, your interstitial fluid, your urine. All right? And we've already done this equation, right? Water plus carbon dioxide, carbonic acid, which dissociates into hydrogen ions, and bicarbonate. What's the enzyme that acts right here? Carbonic anhydrase. Good. All right, and notice the arrows go both ways, so it's reversible. Phosphate and ammonia are important buffers for the urine. Okay, and they're used when we have an excess of acids, so really bad uncontrolled acidosis. And then proteins serve as an intracellular buffer. We're not going to talk a whole lot about that, but just be aware that they can serve as a buffer too because they have charged ends, right? And charged amino acids. All right, we talked about this. Bicarbonate buffer system, most important, minimizes changes in pH to keep it around 7.4. All right, so if we say our body is pH is 7.4, is that neutral? No. It's slightly basic. Okay, it's slightly basic. Why is it slightly basic? Because we have this bicarbonate buffer system. We have a built-in buffer system to control for the acids that are constantly being produced. But we also have our physiological buffer systems. Our lungs and our kidneys can both handle acids in terms of getting rid of them or keeping them in. When would you want to get rid of an acid? When you're in acidosis. When would you want to keep acids? Alkalosis. Good. All right. Just a little bit about concentrations. We're not going to go into too much detail about this. Normal hydrogen ion concentration is about 40 nanomoles per liter. So that doesn't seem like a lot, but that can produce wide changes in pH. Okay? We produce about 60 to 80 millimoles per day. We don't get an acidosis because we're able to buffer that. This is what concentration that equals to in milliliters. What am I doing here? So 47,500 times greater than normal hydrogen ion concentration. That's what we're producing. All right. So just to give you an indication how much acid we're actually producing a day, but we're fine, right? And it's because of these buffering systems. All right. We already said that. A lot of redundancy in this lecture. All right. And then we've already talked about this, right? Our first line of defense, chemical buffer systems. Second line of defense, physiological. All right, so if I were to ask you on a test situation, what is the primary chemical buffer system for our body, your answer would be bicarbonate. And if I ask you what is an example of a physiological buffer system, you would say renal or respiratory. Good. All right. A little bit about volatile acids because that describes how the respiratory buffer works. We know carbon dioxide is an acid, right? The more carbon dioxide we have, the more hydrogen we produce. Okay? The more carbon dioxide, the more hydrogen. So if we are in a situation where, let's say, COPD, where we can't really exhale that much, what's going to happen to our carbon dioxide levels in our body? They're going to go up. And as they go up, what's going to happen to our pH? It goes down. Okay, important thing to remember. If If, let's say, I had like diarrhea all the time, and I'm losing a lot of bases like pancreatic secretions, bicarbonate, I'm losing a lot of bases from my body, what's my condition going to be? Acidosis. So it's the diarrhea causing my acidosis. What's my breathing going to be like to compensate for that? You're going to breathe faster. Why?
Because the less affinity for oxygen.
No, because you're breathing off CO2. You're trying to get rid of that acid. So it's that acid, that increase in acids that causes you to get rid of CO2 to compensate. That's going to be a big term to be familiar with, compensate for that acidosis. Can hyperventilation, that increased rate and depth of breathing, can that cause an acid-base imbalance? Yes. Which imbalance? Alkalosis, because you're getting rid of carbon dioxide. So breathing can be the cause of an acid-base disorder, but it can also fix an acid-base disorder. And write this down. If the respiratory system is the cause of your disorder, the respiratory system can't fix the disorder. So what has to fix it? The kidneys. Good. Okay, so this next slide, we already went through this, so this is just a summary of what we just talked about. And notice that getting rid of carbon dioxide fixes up to 50 to 75% of our acidosis or alkalosis. All right, so those are volatile acids, those we can get rid of from our respiratory system. Non-volatile acids are those that we can't breathe out readily. These are the ones that are produced by our body mostly during metabolism. And what are our cells always doing? Metabolizing. So we're constantly producing carbonic acid, lactic acid, ketone bodies, sulfuric acid, phosphoric acid all the time. And we have to make sure we have a buffering capacity for those systems. Bicarbonate works really well, but our respiratory and our renal system will also help out as well. Okay, but the renal system is more geared towards getting rid of these non-volatile acids. Okay, so they get rid of about 80 millimoles per day. Okay, when we've already talked about glomerular filtration, we filter over 4,000 millimoles of bicarbonate a day. Okay. We secrete from the tubules, we secrete into the tubules 4,400 millimeters of hydrogen a day. So we filter a lot of bicarbonate, so that's in our tubular system. We put a lot of hydrogen in our tubular system. These are pretty close numbers. What do you think they're doing in the tubules? Neutralizing each other, right? Okay. However, we reabsorb most of our bicarbonate. All right, and we're going to talk about the mechanisms of that in a little bit. So even though we filter all that bicarbonate and it neutralizes the acids in our urine, we reabsorb that bicarbonate right back. All right, so we don't lose function of it. All right, but throughout a day, we're also producing new bicarbonates all the time. Okay, we're also producing new bicarbonates all the time. And again, we'll go into those mechanisms in a little bit. Okay, this is just an introductory slide. We're just for the mechanisms we're going to be talking about. We're already talked about we secrete hydrogen ions. We get rid of it. They're buffered by bicarbonate. If we run out of bicarbonate to buffer with, then we start using ammonia and we start using phosphates to buffer. Okay, and we'll go into the details of this in the next few slides. So, how does renal regulation work? Bicarbonate reabsorption, talk about how that happens. Bicarbonate production, we'll talk about how that happens above and beyond the reabsorption, and getting rid of hydrogen ions. So, if I were to ask you how your body compensates for an acidosis, you would say you reabsorb bicarbonate, you produce more bicarbonate, and you secrete acids. Okay, so this is your answer for that right here. All of these are happening all the time. All right, here's your mechanism. All right, we talked about you are filtering through the glomerulus bicarbonate like it's non-discriminate. It's dissolved in the plasma. You're going to filter it. And we are secreting hydrogen. We talked about that. For every bicarbonate that is filtered, it will combine with a secreted hydrogen to form carbonic acid. All right, and of course, on the lumen here, you'd have carbonic anhydrase performing that function. Okay, all right, so we took this hydrogen, we combined it with bicarbonate, we have carbonic acid. So now it's part of a molecule, right? Carbonic anhydrase splits that into carbon dioxide and water. Where does water go? Gets peed out. That carbon dioxide that's produced can then go back into the cells that line the tubule. All right? So this hydrogen right here ended up in this water that we got rid of. So we got rid of the hydrogen, right? And converted it to something that's not going to ruin our kidneys, our tubules. This carbon dioxide that's produced, however, goes back into the cell and will shift back the opposite direction. So carbonic anhydrase is also inside the cell. It will form carbonic acid. That will dissociate, and then we reabsorb that bicarbonate. So what happened? We neutralized the acid here. It went out in the urine. We reformed that bicarbonate and put it right back into the blood. Okay, so it was filtered, but then we reabsorbed it. Is it the same bicarbonate? Not necessarily. But for every filtered bicarbonate, we reabsorb one bicarbonate. And that's how we buffer in the renal tubular cells. All right, and through this process, that is most of our bicarbonate reabsorption. Okay, this is going on all the time right now in us, keeping us out of an acidosis. All right, if we are in such a condition where we're not just secreting a lot of hydrogens, but we're actually filtering them, so we have so much acid in our body, so much hydrogen ions, that we're filtering them and secreting them, and overwhelming bicarbonate, then we start putting new bicarbonate into the blood. All right, so this is a little bit different. The concept is pretty similar, but we're adding new bicarbonate instead of reabsorbing bicarbonate. So here's how that works. We'll come back to this slide. This should actually be afterwards. Okay, here's how that works. All right, remember, we've used up all of our bicarbonate. It's not here anymore. You don't see it, right? So it's not being filtered. It's all used up. But we're still secreting these hydrogen ions. Instead, it will combine with a phosphate, sodium monohydrogen phosphate. You convert that into sodium dihydrogen phosphate, and it goes out in the urine. It's buffered. It goes out in the urine. Okay? What are the two things we didn't produce in this reaction that we did produce with bicarbonate? Water and CO2. Okay? So this is going out instead of the water, but we don't have CO2 coming in, right? So we're not replenishing that carbon source. But this process is still going on. Okay, this CO2 plus H2O plus bicarbonate, or yields carbonic acid. and then that will still put bicarbonates into our system. So since we aren't dealing with any filtered bicarbonate here, but we're still adding bicarbonate to the blood, that's called bicarbonate production. We're adding new bicarbonates instead of reabsorbing what was filtered. Does that make sense? Because we used all that up already.
The CO2 that comes from the cell rather than coming from the tubular.
Right. Through metabolic processes, mostly the Krebs cycle. So when you have CO2 coming back in, that will just add to whatever's already there. All right? So new bicarbonate, net gain of bicarbonate. All right? Same thing happens with ammonia. All right, so there are two mechanisms to produce new. We can either use sodium monohydrogen phosphate or ammonia. So for that secreted hydrogen, we make the ammonium ion. That goes out in the urine. This process right here is still taking place. We're adding a new bicarbonate. All right? So if that secreted hydrogen combines with the bicarbonate, we're reabsorbing bicarbonate. If that secreted hydrogen combines with phosphate or ammonia, we're producing new bicarbonate. Okay? And why would we want to produce new bicarbonate? Right, to compensate for that acidosis, because we have so much acid, it's using up all our bicarbonate, so we're going to have to keep pumping new in. Okay? So this is a summary of what I just said. All right? In an acidosis, where the amount of hydrogen secreted by the kidney greatly exceeds the amount of bicarbonate filtered, we will secrete more protons, which will get excreted. We'll have complete reabsorption of bicarbonate because you used up all your filtered bicarbonate, and we add new bicarbonates to the blood to try to help with that acidosis.
So these all happen at the same time, like the ammonia and the phosphate and the bicarbonate, or is it they choose which they use? Like if we don't have bicarbonate, we use the other two?
Yes, if we don't have bicarbonate, then the other two will take over for it. They will be going out at the same time, but compared to bicarbonate, it's minimal. All right, if you want this in layman's terms, simple terms, how do the kidneys compensate for an acidosis? Reabsorb bicarbonate, produce bicarbonate, get rid of the acid, like we said in that one slide. Okay, these are just some numbers associated with that. I'm not going to go through those. Not that they're not important, don't cross it out, but I'm not going to go through the math associated with that. Basically, it just shows you the math behind that reabsorption. So in the end, the goal is we are adding bicarbonate to the body with a net loss of hydrogen ions at a total of 59 millimolar per day. Bicarbonate. All right. Actually, normal conditions in an acidosis, we can get up to 200 millimole per day. So look at the numbers, 59 versus 200. So we can ramp up our capacity to buffer those acids, is basically what this is trying to show you in numbers. All right. Oops. I hate that it does this. I forgot to get rid of these. Okay, so we know already, if we look at pH, what will that tell us? If we're in an acidosis or an alkalosis. Okay, so that's what the pH tells us. However, we have to know if that acidosis or alkalosis is metabolic or respiratory. All right, how do we know that? How do we know that? Well, first of all, why do we want to know that? Why do we have to know where it's coming from? Right, so we can know how to fix it. Or, because most acid-base disorders are not primary disorders. They're secondary to something else. What did I say the cause of my acidosis was? Do you remember? You guys made nasty faces at it. Diarrhea, right? Secondary problem. Okay, if it's diarrhea, would you call that a metabolic problem or a respiratory problem? Metabolic problem. All right, so the key into knowing whether it's metabolic or respiratory is looking at a particular value. If it's metabolic, we're always looking at bicarbonate. If it's respiratory, we're always looking at CO2. Okay, so if we are in an acidosis, that means we are less than 7.35 pH. Okay? What does that mean in terms of these? If I have a decreased pH, will an increase in bicarbonate cause a decrease in pH? No. So can metabolic be the problem if it's up? No. But if we decrease bases, can that decrease our pH? Yes. So if we see a decrease in bicarbonate, that would be a cause of metabolic acidosis. All right? Why am I going through this in painstaking detail? Because people fuck that up all the time. Okay? That's a really difficult concept usually for people to wrap their head around. So if you wrap your head around that, you're good. Respiratory is easy. Respiratory, CO2 is a what? Acid. If we increase acids, will that make things more acidic? Yeah, so an increase in CO2 will give us a respiratory acidosis. But if we decrease CO2, will that make things acidic? No. So it can't be a respiratory acidosis. So I put it to you, if we had a metabolic acidosis where CO2 is also decreasing, why is CO2 decreasing? Good, to compensate. All right, so the problems we're going to do at the end of all this is knowing how to determine what the cause of the problem is and which one's trying to fix it. Okay? Similar concept for alkalosis. All right, if we increase bicarbonate, that can lead to an alkalosis. If we decrease CO2 or decrease acids, that can also lead to an alkalosis. If you get screwed up in that process, this is a nice flow diagram to tell you how to interpret acid-base problems. Okay? So look at the pH first, then look at which one is matching the pH, which one could possibly cause that pH change. All right, what can cause these acid-base disorders? Okay, what can cause them? So a metabolic acidosis, again, remind me, which value are we looking at? Bicarbonate. And what's it doing? Dropping for metabolic acidosis. All right, aspirin poisoning. Ketoacidosis from diabetes. We already mentioned diarrhea, right? You guys won't forget that, hopefully. You will, but that's okay. Problems with the kidneys, renal tubular acidosis, or certain drugs will also do it, carbonic anhydrase inhibitors. If we have carbonic anhydrase inhibitors, are we reabsorbing bicarbonate? No. So things are going to get more acidic as a result. In fact, this can lead to a renal tubular acidosis.
What do people take those for?
Carbonic anhydrase inhibitors?
Yeah. And what's the benefit of taking something like that? Is that a farm question? Sorry.
That's a farm question.
Okay.
Possibly an alkalosis. Like, think about it. If you're in an alkalosis and you don't want to reabsorb bicarbonate, that might help with that. But there's probably other reasons why you want to take it too. That one I'm not familiar with. All right, respiratory acidosis. What can cause that? All right, so if you have a respiratory acidosis, what does that mean you're doing to CO2 in your body? You're holding on to it, all right? So what kind of things will reduce your breathing? Think about that. brain damage, especially to the pneumotaxic area, pneumonia, emphysema, other lung disorders, opiate overdose. Metabolic alkalosis, all right? What kind of things other than the lungs can cause you to be more alkaline? Anybody know people who like just pop Tums like they're candy? You do?
No. At one point.
I know people. My brother used to do this all the time. It was, at the time, I didn't know any of this stuff. But just used to pop them like candy. No issues, but potential issue could happen. Vomiting gastric acid. All right, so if I feel sick right now and I go to the bathroom and I throw up, I lose acid, right? Is that going to put me into an alkalosis? No. But if I throw up like every couple hours all day for a couple days, that will cause me to lose acids, okay? So that could lead to an alkalosis. Mineralocorticoid excess. What's our mineralocorticoid? Do you guys know? Mineralocorticoid? Aldosterone. Because aldosterone can also lead to getting rid of hydrogen ions as well as potassium. And overuse of diuretics. And that's related to potassium balance. All right, respiratory alkalosis. What kind of things can cause us to get rid of too much CO2? Anything that's going to make us go fear, high altitudes, pain, all of those. So these are all the things that can lead to these. So telling you that acid-base problems are a secondary disorder to these things we just mentioned. Okay, so have a good feeling of things that could happen in your body that could lead to these things. If it's a respiratory problem, it's always going to deal with the lungs. If it's a metabolic problem, does it always have to do with the kidneys causing the problem? No. Metabolic means everything but the lungs, okay, not just the kidneys. Oh my God. So, it's so stupid. All right, this is a summary table of all of that. Let's go through one of these just to show you what happens. If we're in a metabolic acidosis, okay, of course we're going to see a drop in pH. That's going to tell us that, let's say, 7.30. I know that looks like a 2. It's a 7. We see a decrease in bicarbonate. Can a decrease in bicarbonate cause an acidosis? Yes, so that's how we know that's the problem. Why do we see a decrease in CO2? To compensate, okay? So CO2 is decreasing to compensate. So the compensation, what we're going to see in our patient is to compensate for that disorder. And we're going to see an increase in bicarbonate production, all right? So if it's a metabolic problem, we see a respiratory compensation and we see a renal compensation. What do you notice about respiratory acidosis and respiratory alkalosis? You don't see the ventilation, all right? Because if respiratory is the cause of the problem, respiratory cannot fix the problem. Good. So, really good summary slide. I will put it to you, when we do acid-base problems today, yes, we'll do them. And I forgot to print out sheets for you, but they are in Canvas for you for practice. So if you guys want to pull those out on your devices, which I think is awesome. You guys got to teach me how to use those one time. I had a student from cohort 23 come to me yesterday. Like, it's neat because all 3 cohorts are on campus this week. Really cool. And students are...
Why are they on campus?
End of rotation exams. And today they're taking their PACRAT 2 as well. So it's really... I love when I see students on campus doing student things. They like... one of the students was walking down the hallway going, "Nobody knows the trouble." And I'm just like, you're crazy. So I love that. But one of my advisees came in and talked to me yesterday. And she said, "Oh, it was your birthday? Happy birthday. Are you 50 yet?" Like, fuck you.
Are we going to check the calculations on the exam? Are we doing any, because remember how you said, to your knowledge from last week, that there was no math on the test? Is that going to change? Are we have to do
There will be no math, but there will be acid-base problems.
Okay.
It's mathematically associated, but it's not actual math, okay? So, when we do those problems, what you're going to see in some of them is a change in pH and a change in one of these values, but not a change in the other. That almost never happens, okay? Doing those is going to be a kind of practice to get us into how these are set up. Has anybody done these before? That's why she's looking at me like, I know this, yeah. Okay.
This was like a big part of my pathophys class.
Okay. So I taught this in Pathophys as well. We teach it in AMP2, Pathophys, because apparently the nursing instructors don't do a good job at it, so they make the science people bone everybody up on it. It was funny when I was taking nursing classes, and they were teaching this. The same guy who came in here and yelled at you was teaching that part of the class. And he'd always go to me, "That's right, right?" And I'm like, "You're fine. Go. Don't point me out anymore." Okay, so this is just another, I have a lot of redundancy in this PowerPoint because there are multiple ways to look at the same thing. So this is telling you the same thing that this is telling you, only in a flow diagram. All right, so if you have trouble with it, this is another way to go through and see what's going on. So I'm not going to cover it in any more detail than what I already have. So these are here for you. Okay, now we get to the acid-base problems. All right, the primary objective is to get back within 7.35 to 7.45. So what we're going to see are three things. We're going to see a pH. Still no? Oh. There? Okay. We're going to see a pH. We're going to see a PCO2. And we're going to see a bicarbonate. Normal values for pH should be what? Good. Normal CO2 should be, oh crap, 35 to 45 millimeters of Hg, millimeters of mercury. And bicarbonate should be, you look in 5 different books and 5 different hospitals, you're going to get 5 different values. Normally what I teach is 22 to 26, but I've seen it as far as 21 to 28 milliequivalents per liter. So let's go with this one, since it's a wider range. So anything below that, or anything below 21, or anything above 28. Okay?
What's the unit for that?
Milliequivalents per liter. What's the units for pH? There are none. Good. So I will ask you plenty of trick questions in class. I won't ask trick questions on your exam. I promise. That I think that's really shitty to do to students. So I will never do that. I hate it when people did it to me, so I won't do it to you. Fuck the system. All right. What I won't do is give you normal values. So you're responsible for knowing them.
We're going with 21 and 28.
21 to 28, since it covers a wider range. Okay. All right, pH is 7.49. CO2 is 31. Bicarbonate is 24. Okay, how do we go about this problem? First, we look at the pH. What's pH tell us? Alkalosis. All right, so we have half the problem. What am I doing? Where did I put in my pocket? What's the acid-base disorder? Okay, we know it's an alkalosis so far. CO2, is it high, low, or normal? Low. Bicarbonate, high, low, or normal? Normal. Okay, so if that's normal, could that be the cause of the problem? No. So by default, CO2 is the cause of the problem. So what do we have? Respiratory alkalosis. Okay, so what's the acid-base disorder? Respiratory alkalosis. Has compensation started to occur? No. How do we know that? Because bicarb is normal. Okay, so bicarb is normal. No compensation. Okay, if compensation were to occur, what would we see here happen?
Increase in
We would see a decrease in bicarb. Okay, so maybe we would see a 19. Okay, what would happen if we increase bicarb? That would make things more alkalosis. Okay, so we want to decrease bicarb. How are we decreasing bicarb? Less reabsorption. We're not reabsorbing, and we're holding on to our hydrogens. All right, we're still going to filter bicarbonate. We can't control that, but we're not going to reabsorb it. Okay, so we're not going to reabsorb bicarbonate, and we're not going to be secreting hydrogens.
How do we control like the reabsorption rate of bicarb? You might have said it, but it's also acid.
No, it would be through. So if we're in an alkalosis state, we're going to have an excess of bicarbonate. We're not going to be. We're still going to be reabsorbing some, but a lot of it's going to go out in the urine too. So.
Like more because we reabsorb 90 to 95%, right?
Right.
So we would have less reabsorption, like 80 or something.
Yeah, we'd have less. Well, we'd have less reabsorption because we're buffering less acids. We don't have as much acid, so we can't buffer it, and so it's going to go out in the urine.
That makes sense.
Okay, good.
Would we be able to ever know like which type of buffering is going on for that? Or no? Like how you're going through the different pathways when we're not reabsorbing or secreting?
It's all the same.
Okay.
In this case, we won't have any of that stuff that we talked about because this is an alkalosis. All right. What was I going to say about this? Does that make sense, though?
Yeah.
At least on a surface level. All right, let's do another one. Notice I gave you a harder one right away. The easier one would have been giving you an increase in CO2. I'm sorry, an increase in bicarbonate. All right. pH is 7.31. CO2 is 48. Bicarbonate is 30. What do we have going on? Acidosis. Respiratory and metabolic. This is increased, and this is increased, right?
Yeah.
So which one's causing the problem? Respiratory. Why?
Because bicarb too.
Go ahead.
Because bicarb is elevated to compensate.
Okay, but how do we know that's not causing the problem?
If there's an increase in CO2, you know it's respiratory.
How? Why?
Because acid.
Carbon dioxide's acid, right? Carbon dioxide's acid. So if you increase acids, you're going to make things more acidic.
Yes.
That's what I was looking for. Is that too basic? Like, I try to make sure. I try to scoop up everybody along the way. Okay, so we know it's a respiratory acidosis. Is compensation occurring? Yes, compensation is occurring. But is our pH out of normal range? Yes. So what we would actually call this is a partial compensation. Okay? When would it be complete or full compensation? When we're back to normal range. So what would be my pH? It would be 7.35. Could it be 7.45? No. Because if you're just getting back from an acidosis, you're not likely to go all the way to the other end of the spectrum. Okay? Let's do that. Oh, so this acidosis, how are we increasing our bicarbonate? Production, reabsorption, and secreting protons. Good. All right. pH 7.28. Bicarb is 18. CO2 is 30. What do we? Oh, damn. So if we look at the pH, what do we know? If we just look at pH, we're normal. Do we agree with that? Okay. What do we know about CO2? It's low. What do we know about bicarbonate? It's low. Okay. Both of those are low, but we have a normal pH. So what does that tell you? Complete compensation. So we know the end of the story before we know the beginning. Complete compensation or full compensation. Okay? All right, what is this pH closer to? An acidosis or an alkalosis? Acidosis. So what is it more likely we recovered from? An acidosis. So we had an acidosis. Which is the cause of our problem? Metabolic. Decreased bases makes things acidic. So we have a metabolic acidosis. Why is CO2 decreased though? It's compensating. So that's why we have the complete compensation. What is our respiratory system doing to compensate for this? Increasing rate and depth of breathing. So you see a patient who is hyperventilating, and that's all you know about the patient. What's your first thought? Really? Couldn't he be in an alkalosis because he's hyperventilating? The answer is you don't know. You need more information. Okay? So you just see a hyperventilating patient, you got to dig a little bit deeper. All right? Does this make sense? Okay. So where are we on time? 8:53. Yes. I did good on that one. You guys ever seen, I know you guys are young, you guys ever seen the old Nutty Professor where the little kid's like, Hercules, Hercules? It's been a long time. All right, so these, I promise you, will be on your exam. Okay? It's a 100% guarantee. We dedicated an hour to it. You should be able to go through problems like we just did here. This was probably one of the more complicated problems you'll come across. Okay? And that's as hard as it gets. So for those acid-base problems, they're in Canvas. There's set 1, set 2, set 3. So you don't have to do all of them. Do as many as you feel to be comfortable. Okay? I will post keys for them so you know the answers either tonight or sometime this weekend. Okay? So that ends acid-base. Not that bad, right? Okay. All right. Let's take a break there until...
You want to do 9?
Yeah, let's just do till 9. All right. So this is all about your IOs originally had like some cardiac muscle stuff in here and some smooth muscle stuff. I took that out and just focused on skeletal muscle because most of it's similar enough to where you get the gist. And I did do the research, and no board question is going to ask you about physiology of smooth muscles. It's going to be more clinically related. So we'll talk about skeletal muscle structure, which you guys have been looking at with Victor and Victoria for the past 2 weeks. So this is kind of what is skeletal muscle and how does it work. So note that skeletal muscle is not just muscle tissue. We also have nervous tissue to make sure it works, blood to make sure it has nutrients and gets rid of waste, which is why you have to know nerve supply and blood supply to these tissues in anatomy, and a lot of connective tissue. All right, so we know that muscles are covered in fascia, right? And that kind of holds things in place. And what connect? Couple terms. What connects muscle to bone? Tendons. What connects bone to bone? Ligaments. What connects muscle to muscle? Good. Excellent. Oh, it's right here too, I guess. Interesting thing about tendons is tendons are made up of dense, regular connective tissue, but they are literally interwoven into your bone, which is why they have such a high tensile strength on the bone. Like you can lift a lot of weight before actually pulling these tendons off of your bone because they are literally part of the bone. They are interwoven in between them. Right? To start off, we're going to talk about musculotendon reflexes. So there are 2 types of reflexes related to your muscles. One is caused by this thing called the Golgi tendon apparatus. All right. So in your tendon, there are these receptors, and they detect any change in tension on the tendon. Tension on the tendon. So that's a good way to remember it, right? Tension, tendon. As the muscle contracts. So a way to think about this is, let's say you have something heavy on the ground and you're lifting it, and you have a lot of tension on that muscle, right? Okay? What happens is, as there's tension, it's going to send a signal down a sensory nerve fiber. These are called 1B nerve fibers. Into the dorsal portion of the spinal cord. This is going to come up later, but all sensory goes in through the back of the spinal cord, through the posterior portion. Okay, all sensory through the back, all motor out the front. All right, so it's going to go in. Whenever we have a synapse and there's a plus next to it, what does that mean? Whenever you have one neuron synapsing onto another neuron and there's a plus there, what do you think that means? Begins with A, rhymes with schnactimation. Good. Activation. I make shit up, okay. All right. There's this interneuron between them. That is called an interneuron. All right, and it's the interneuron that allows flexibility of the signal. And that's going to synapse onto a motor neuron. These are alpha motor neurons. So these are your big motor neurons that go to extrafusal muscle fibers and cause them to contract. Extrafusal muscle fibers are most of the fibers in your muscle. Okay? So what this interneuron does is it synapses onto this alpha motor neuron. What does that negative sign mean? It inhibits. All right? So you have a lot of tension on your tendon from lifting up like this, And then the signal pathway detects that tension and inhibits your muscles. What are your muscles going to do? Why?
Cowardice. Sorry. It's a safety mechanism.
Yes. You can't lift that, you're a bitch. I mean that in a very white trash way, not in a derogatory way towards anybody. Disclaimer. Okay, so it protects. You don't want that tendon popping off the bone. Mike Whitmer down here runs the science labs. Really big. You guys ever seen him? Really big guy. He has hair kind of combed back, kind of like mine. But he's if you've never seen him, if you do see him, you'll know who Mike is. I've known him for a long time. He was lifting a lot at one point in time. Tore his bicep right off his shoulder. Because overdid it. Okay. This protects against you doing that. Okay, so if you're at the gym and you're lifting and then all of a sudden your muscles give out, it's because your body's saying, douchebag, you're not ready for this yet. All right, so protective mechanism. All right, the other one you have is a muscle spindle reflex. All right, this one responds to stretch. All right, when are your muscles stretching? When you're stretching. I'll give you two good examples of that. One is when you jump off of, like, let's say a table onto the floor, the tendons or your muscles stretch, and that causes you to activate certain muscles so that you don't lose balance once you jump down. Okay? The other place you'll see this is you did it with Professor Gavin. You're sitting on a table and you go whack to the front of the knee. That stretches muscle. All right? It stretches these intrafusal muscle fibers. So intrafusal muscle fibers are these little smaller sets of fibers that have a specialized function to get you to contract. Okay? So Jesse's sitting here, and I go smack him with a hammer. That tendon, or the hammer hits the tendon, pulls that muscle just a little bit, right? It doesn't pull it much to activate this thing. And what does his leg do? Kicks out, right? So you activated this muscle to contract. So when this spindle is stretched, sends a signal down 1A and 2 nerve fibers, okay? They synapse directly to a motor neuron. When we have a synapse directly to a motor neuron without an interneuron in the way, we refer to that as a monosynaptic reflex because there's only one synapse in the pathway. All right, the type 2 fibers will activate extrafusal muscle fibers, and that's why he goes, okay? But what do you think is going to happen to, let's assume this is like a spring. This green thing is like a spring. What do you think is going to happen to that spring as the muscle contracts? It's going to shorten. Actually, the spindle is going to shorten, but what's going to happen to that spring that's surrounding the shortening spindle? It's going to go slack, right? So what the type 1A fiber does is it makes sure that the whole unit contracts so that it maintains its shape. Because if you contract your muscle, but that spindle remains stretched out, it's going to be detected as a stretch and oppose your contraction. So you want to make sure that spindle apparatus contracts with the extrafusal muscle fibers to maintain a unit. Does that make sense? Kinda? A little bit, maybe? These coils surrounding the spindle, the sensory coils. So if you contract the spindle and it gets fatter, those coils kind of unravel a little bit. They would unravel. So what this does is it allows the coils to go with the fiber and maintain the shape so it doesn't appear as stretched while the muscle is contracting. So the whole thing contracts, not just the extrafusal muscle fibers. So why do we have this? Proprioception. Do you guys know what that is? No? Go ahead. Say it. I'm just going to stand here. Jerk. My son yesterday, I got to tell you this story. He's always worried about how much money we're spending. It's the weirdest thing. So we went out last night to the store, came back, and we got Frosties along the way, Wendy's. And he's like, I don't want one. I'm like, we got you one. You're having a Frosty. So we get home, we go into the house. He's like, I don't want one. I wanted him to carry his Frosty, and he wouldn't do it. I said, all right, I'm going to dump it. He's like, I don't care, dump it. I said, that's going to waste all the money I spent on this. And he's like, fine, Dad. Because he's so worried about us wasting money. Weird kid. Where was I? All right. Excuse me. Skeletal muscle organization. What you see on a body, like let's take that leg. Actually, did I tell you her nickname is Betty? I nicknamed her Betty. I don't know why. I think the student and I who dissected it came up with that we were going to call her Betty. What you see on Betty is the muscle. But you know all those stringy things that are kind of flaking off at the moment? I know. Shut up. Those would be fascicles of that muscle. So all of the lines you see in a muscle would be the fascicles of a muscle. So little bundles of muscle fibers. And then each one of these fascicles is made up of a bundle of muscle fibers. Each one of these fascicles has bundles of muscle fibers, and these muscle fibers have your contractile proteins. All right, so we're going from a macro level looking at the muscle, and then it's dividing down into the molecular level. Okay, so all of these contractile proteins build up to make this big muscle. That's kind of how it's organized, and this is a flow diagram kind of showing you that what the picture's saying. All right, what we're concerned about is this level, because what happens at this level translates to the bigger level, and we're going to try to build an argument for why that is. All right, surrounding this muscle fiber, you have a network of ER, endoplasmic reticulum-like structure that's called the sarcoplasmic reticulum. Okay, and just outside of that, you have a cell membrane. All right, the cell membrane of a muscle fiber. Jesus, my brain is just went. It's called a sarcolemma. Yes, sarcolemma. All right, and the cytoplasm within that cell membrane that's surrounding everything is called the sarcoplasm. All right, and that's surrounding the sarcoplasmic reticulum and every one of these muscle fibers. Just that proteins are packed tightly together, so it has a little bit different organization than a normal muscle, a normal cell. Okay. If we look down at the myofibril level, these contractile proteins, we know that they are myosin and actin. In fact, we've talked about this when we talked about, clotting, right? We have myosin and actin in what blood cell or cell part? What? Platelets. Good. Okay. When we look at a muscle fiber, when we look at muscle under the microscope, we see these alternating patterns of light and dark lines. What do we refer to those as? Striations. All right. If we look a little bit deeper, so this would be an electron micrograph that's falsely colored, of course. Here would be your light, and here would be your dark. All right, so alternating light and dark. So that alternating light and dark is not organized by light and dark, but it's organized between these blue lines right here. And this whole thing is called a sarcomere. All right, and a sarcomere is the contractile unit of a muscle. Sarcomere is the contractile unit of a muscle. Did you guys see where I said that? I threw it away. Never mind. All right, so if we take this down a little ways and look at like a cartoon of what's happening at the molecular level, the light bands here, they're made up of actin. This picture doesn't show it accurately, but those are made of actin. The dark bands are made up of mostly myosin. Okay, and we'll go into a little more detail about how that's organized in the next few slides. So, we're going to go back and forth between like a conceptual level and a molecular level. When we look at the striations, these light bands right here, remember I said that this blue line right here represents sarcomeres, but each sarcomere, each I band represents only part of one sarcomere and part of another. Does that make sense? Okay, so we call those light bands I bands, and they are made up of strictly actin. So you notice the actin right here in blue. That's the I band. Okay? The actin is attached to Z lines. So you have these Z lines. You have some actin going towards one sarcomere, and then on the other side of the Z line, you have actin going towards the other sarcomere. All right, so that's kind of how it's organized. All right? The A bands are dark bands, and of course I have like these little things to help remember. A bands are dark bands. Now you notice with the dark bands, there's really dark and there's kind of dark. All right? The kind of dark, that's called the H zone. That's made up of strictly myosin, only myosin. All right? But where it gets darker, you have to assume that you have multiple overlapping proteins. So the dark bands at the end, that's actin and myosin overlapping with each other. All right, so that's what's making up these bands that we see under the microscope, these striations. Okay, so that's showing you the big picture of what it is. So here's your A band. Just looking at this, what is this portion called? H zone, and what's it made of? Myosin. These darker portions are made up of actin and myosin, and you can see that right here. And then the I band is made up of strictly actin. Okay? And they're organized so that myosin sits in the middle of two opposing actin filaments. All right? And what's going to happen is myosin has little heads on them that go like that cat in the Chinese restaurants that I don't know what that's called. Actually, I have a good friend who used to teach physics here, and he loves cats. But he always makes fun of people who are not as intelligent by saying that they are like cats playing with a ball. It reminds me of that, too. Does that make sense? Oh, what I was going to say. You got this going on. Attaching to the actin, and you're going to pull them towards the center of the myosin. Okay, so basically the myosin is going to pull the actin towards itself. And what's that going to do to the overall sarcomere? It's going to shorten it. Okay, so that's the basis for how we do a contraction at the molecular level. So what we're going to talk about are the events that happen in this area and then translate that into a broader muscle, how that translates into broader muscle contraction. All right. Now, surrounding these, you have that sarcoplasmic reticulum that we mentioned before. So sarcoplasmic reticulum is much like endoplasmic reticulum, except that its primary function is to store calcium. All right, so you have a scientifically termed a shitload of calcium inside of the sarcoplasmic reticulum because it's a storehouse. And then attached to those are these things called transverse tubules. Okay? And transverse tubules are connected to the sarcolemma. What's the sarcolemma again? The muscle cell membrane. Good. So that when the muscle cell membrane depolarizes by a motor neuron, that signal gets translated down the transverse tubule and opens up calcium channels in the sarcoplasmic reticulum. All right, now tell me, if I have a bunch of calcium inside of this tube right here, and I all of a sudden open channels, what's that calcium going to do? It's not going to go into blood. It's going to go out into the cell. Okay? So think of this as a bag inside of a cell full of calcium. You open that bag, the calcium goes into the cell. Okay? How would we get calcium back into there? As our friend on Wednesday said, you pump. There are calcium pumps that pump calcium back in. Okay? And that's how we restore the gradient. All right, so let's see how all of this happens. Much like cardiac, in order for a muscle to contract, you have to have what kind of signal to get it to contract? Pardon? Electrical signal, right? And that has to come from the nervous system. So we're going to talk about the neuromuscular junction, where a motor neuron contacts the muscle cell membrane. Okay? And that is a modified, of course, synapse. Normally think of synapse as an axon with a dendrite, but this is an axon with a sarcolemma. And where the axon contacts, the point of the sarcolemma where the axons contact is called the motor end plate, and that's strictly a muscle cell phenomenon, the motor end plate, whereas the neuromuscular junction is both the axon terminal and the muscle cell membrane. All right? All neurotransmitter coming from the motor neuron to the sarcolemma is acetylcholine. Okay, all of it's acetylcholine. So, how do we use this to stimulate a muscle to contract? All right, so I'm going to draw my cheap-ass neuron. We have an action potential coming down. Do you guys remember the process of this? When an action potential reaches the terminal, you open up what kind of channels? Good. Voltage-gated calcium channels. This is review. You'll find this nowhere in the PowerPoint. This is just me kind of going back to the asynchronous weeks. Calcium rushes in, and what does that cause? Vesicles to fuse with the presynaptic membrane and release a acetylcholine. Okay? If you've ever taken cell biology, it's way more complicated than that. Not the point of this lecture. Okay? So acetylcholine is released from synaptic vesicles into that, synaptic cleft. Okay, so we're right here. All right. Why do we have neurotransmitters? What do they always do in every case, no matter what? Bind. They bind to receptors. All right. So it binds to acetylcholine receptors on the sarcolemma, and almost always when a neurotransmitter binds to a receptor, what happens? Be more specific. How? How?
Conformational change of a receptor.
Which does what?
GPCR.
No. Sometimes, if you're talking muscarinic. Oh, come on. Say that again? You open up sodium channels. Oh, you open up some kind of channel. Here it just happens to be sodium. Okay? So you have these receptors in the membrane. They will bind, or acetylcholine will bind. That will open up the channel, and sodium rushes in. And that sodium rushing in is the basis for, Maddie? Or who said it? Did you say it? Depolarization? Who said depolarization?
Me.
You said depolarization. It's the basis for depolarization. I didn't mean to put you on the spot. I thought you said depolarization. Okay? So that's where we are right now. Does that make sense? Okay. So it looks a little bit different than what we put previously, but the same concept arises, or same concept applies. You have these dense bodies near the presynaptic membrane. These dense bodies, what they're thought to do, they're not 100% sure, is to orient these vesicles so they get secreted in a very specific way. Okay? So the vesicles fuse. You have all of these, I'm going to put it to you that even though this is lacking, these blue ones right here, they are over here. Okay? So I'm going to use, let's do this. I'm going to use the one on the left as my example because there's more room. All right? So we have acetylcholine bind to these receptors. You open them up, sodium rushes in, right? So you take a polarized membrane, let's say minus 80, and you start taking it down to what? 79, 78, 77. Okay? If enough acetylcholine binds to enough receptors and enough sodium rushes in, that depolarization spreads down to these voltage-gated sodium channels. Okay? And when these voltage-gated sodium channels detect a threshold potential of, I think in a couple of slides it shows it as minus 40. Doesn't matter. When they get to threshold, what's going to happen? All of these open, and then what happens? A shitload of sodium rushes in, like a whole bunch of sodium rushes in, and that's what gives you your what? Your what? Action potential. Good. So that's what's going to create the action potential in the sarcolemma. All right, so we talked about 1 and 2. I tried to help you guys keep track by, using blue for new stuff. Sarcolemma depolarizes. You generate the muscle impulse. Another name for the muscle impulse is what you just said, action potential. Okay, that's going to travel across the entire muscle fiber. But then what did we say is attached to the muscle fiber? T-tubules, right? Remember, T-tubules are attached to the muscle fiber. and those are going to depolarize. Okay? So this is just showing you the action potential. What this graph says is here is your resting membrane potential. You open up those, acetylcholine opens up channels. You get a depolarization. Why would it go back down? You didn't depolarize enough. You didn't activate those voltage gated channels. So that's why it stayed down here. Is there more? No. Okay? So if you open up enough, then you go above threshold and you fire that action potential. Okay? I know a lot of people miss this. This phase of the action potential, high permeability to sodium. This part of the action potential, high permeability to potassium. Yay. Okay. All right. It was, okay, what started this process? Acetylcholine. Okay. If you want to shut off this process, you have something that breaks down the acetylcholine, and that's called acetylcholine ester ACE. Right? ACE means it's a what? Enzyme. All right, that breaks down acetylcholine. All right, here's how this works. In the synapse, right here, you have acetylcholine. Let's use this green as acetylcholine esterase. That will break acetylcholine down into acetyl and choline. The acetyl group diffuses off somewhere else. Choline gets reuptaked. It goes back into the presynaptic terminal. So that's how you can recycle acetylcholine. Then it attaches an acetyl group to it and package it into vesicles again. All right? As long as acetylcholine esterase is there, you're breaking down acetylcholine and stopping the signal. This is normal. This happens all the time. This is how we stop action potentials. Why do we want acetylcholine in our synaptic cleft?
Because then the action potential will keep going and you can't get relaxation.
Right, because you keep stimulating the muscle to contract and you never shut it off. So we need acetylcholine esterase to turn that off. Okay, that's an important concept because if something goes wrong with acetylcholine esterase, what does that create? Okay. So here's what your action potential looks like. Resting potential, not polarized enough. Here's what your action potential looks like in the graph. There are certain drugs that can modulate this. Okay, one of the drug inhibitors specifically are curariform drugs. What they do is they block acetylcholine channels. Okay, so here's your neuron. Here's your sarcolemma. Here's your acetylcholine receptor. What curariform drugs do is they will block acetylcholine from binding. If you block acetylcholine from binding, what don't you do here? Right, you don't open up the channel, and you never have sodium going in, so you never depolarize the membrane. Okay, so that's one way that you can cause. Oh, let me ask you, what would this do to a person? You give this drug, they don't get action potentials, so they never contract their muscles. What's too many at once? What'd you say? No, that's long term. Immediately, you get the drug paralysis. So these are paralytics. All right. Another drug is botulinum toxin. All right. What that does is it decreases the release of acetylcholine from nerve terminals. So you never have acetylcholine being released from the motor neuron. If you never have acetylcholine being released, it never binds. If it never binds, you never open the channels. If you never open the channels, you never depolarize. Okay, so you're not going to contract. Then we have stimulants. All right, stimulants of end plate potential include acetylcholine-like drugs. So if you want to stimulate muscles for whatever reason, these kind of drugs you would give, these are the names of some of them, nicotine included, they will activate nicotine acetylcholine receptors because you have a man-made drug that mimics acetylcholine. So of course it's going to open up receptors. If you open up receptors, you're going to open up channels, and these are not destroyed by acetylcholinesterase. So what does that mean?
It's getting harder for the contractions.
Right, the signal keeps going. Okay, another drug you could give as a stimulant for muscle contraction would be anti-acetylcholinesterase. All right, what anti-acetylcholinesterase would do would be what? Inhibit acetylcholinesterase. Acetylcholine stays in the nerve terminal and continuously activates. So this is very similar. You've probably seen commercials or heard of this in maybe neuro lectures where you have drugs, SSRIs, for example, or SNRIs, that prevent reuptake into the presynaptic terminal, and that floods the synaptic cleft with neurotransmitter. This is the same concept. All right. Dangerous chemical, sarin. You guys ever heard of this one before? Mm-hmm. Some of you have. Okay, sarin, nerve gas, inhibitor of acetylcholinesterase, and notice that it raises acetylcholine to 26 times, acetylcholine levels to such high levels that makes it more deadly than cyanide. And we know cyanide is a deadly poison because it was used inappropriately in certain times of war, right? What sarin does is it binds to a particular site on acetylcholinesterase, renders it inactive, and usually we have death because of asphyxia. So how would sarin inhibiting acetylcholinesterase result in asphyxia?
Your lung muscles.
Say what?
Your lung muscles can't.
How about your respiratory muscles?
Oh, yeah. Right.
Your respiratory muscles. Which one in particular? Diaphragm, external intercostals. Wouldn't contract. You wouldn't breathe, so you die from asphyxia. All right. Treatment, atropine, and this other drug, pralidoxime. All right. Ooh, you guys notice this?
Yeah.
Sorry, I didn't catch that. What this does is it reactivates acetylcholinesterase. All right, so this drug will reactivate acetylcholinesterase, so you start clearing the synapses. Then atropine will block muscarinic acetylcholine receptors. All right, the receptor for acetylcholine at the neuromuscular junction is a nicotinic acetylcholine receptor. Atropine blocks muscarinic acetylcholine receptors, which are not in the neuromuscular junction, but they still are effective. Why? Because those receptors are in the CNS. Okay? And they will block CNS and autonomic nervous system effects, preventing seizures.
Can you say that again? The muscarinic versus nicotinic.
Which
Where
part?
they are.
Where they are. Nicotinic at the neuromuscular junction. Muscarinic in the central nervous system, and specifically the parasympathetic nervous system. And they'll reduce neuron activity. Okay? So going back, so that's just a segue into some drugs that will affect the neuromuscular junction. Let's go back into the contraction. Where are we in time? Dang, I'm doing bad now. Actually, this doesn't. Are we? Oh, okay. This won't take long. All right. What these represent is your action potential in the sarcolemma. Here you have the action potential traveling down the transverse tubule, and here you have it activating the sarcoplasmic reticulum to let calcium out. So that's what this is showing you. And then, of course, when this is no longer stimulated, you pump in calcium back into the sarcoplasmic reticulum to restore the gradient. So you can do this all over again. Okay? Nice try. We're almost done. Yeah, there are. And that's where the effects of smoking will take place. All right, so specifically what happens, let's assume this is a transverse, what the hell? A transverse tubule. Okay, it depolarizes. There's a receptor called the DHP receptor. I don't care that you know what the name of it is, what DHP stands for. But what it does is it's attached to channels on the sarcoplasmic reticulum. And when you depolarize, this will act like a spring and pull the channel open in the sarcoplasmic reticulum, and calcium will start flooding out. Kind of a neat setup, I think. Then when you repolarize the membrane, of course, that channel will snap shut. No more calcium will leave, and these pumps will dominate and pump calcium back into the sarcoplasmic reticulum. So let me ask you this. This calcium pump that's pumping in, does that stop during depolarization? No, it's continuously going. It's continuously pumping in. It's just that the amount that is leaving overwhelms what's able to pump in, and that's how calcium will leave. Apparently I lost control. Sometimes. Did that change? Okay. So, we have these calcium ions that are out of the sarcoplasmic reticulum, and they flooded the cell. So your cell is your nerve, muscle fiber, is flooded with calcium. Okay? It's out there. Here's your contractile proteins. What's this one right here? Myosin. Thick filament is myosin, so this would be actin. Okay? Covering it, you have two proteins. You have troponin and tropomyosin. The purpose of the calcium is to bind to troponin, and it moves tropomyosin out of the way. Okay? The goal, you want myosin to interact with actin, but the sites for myosin on actin are covered. So what calcium does is it helps uncover those actin or myosin binding sites. So if you look here, here's the tropomyosin, and if you look here, you see all these purple dots that represent the binding sites now uncovered. So as soon as you uncover a binding site for myosin, what's myosin going to do? It's kind of greedy. All right? So myosin's going to bind. All right? What did I say myosin does? Remember? Yep. You guys won't forget it. What am I doing? Okay. All right. When myosin binds, I want you to notice the position. The position is it's cocked back. Okay? If it's cocked back, what does that mean it can do? All right? So it's cocked back, it binds. What's attached to myosin? Look at the picture. What's attached to myosin? Besides actin? ADP and phosphate. What does that mean? No. What does that mean happened previously? No. What gives you ADP plus PI? ATP breakdown of ATP, right? So it's the ATP breakdown that allowed it to get cocked back, that provided energy to cock it back. So think of those, like go back to Lord of the Rings. Think of the trebuchets and the catapults. It takes energy to cock them back, right? And then you just release and bing. Okay, so it binds. You have ADP and phosphate that cocked it back. Then they move forward. So this is them moving. So notice they go from this position to this position. So they've moved forward. And when they are attached to actin, they're going to move actin forward as well, right? Okay. When they do that, ADP and phosphate fall off. So they're no longer attached. What does that mean? That means what can attach now? ATP. Good. All right? As soon as ATP attaches, what happened here? They released. Okay? So you're cocked back with ADP. You attach. You release. Move forward one unit. ATP binds. You let go. Okay? ATP is then hydrolyzed. What happens? You guys ever seen those videos? They're stupid. Where they show these people in like the slingshot ride that they pull back and just fling you up into the air and like so you're like in the slingshot mode, ready like. And then as soon as it's hydrolyzed, it can do what? Rebind, detach, or. And actually, if you go out and Google and look at videos of muscle contraction at the molecular level, do all kind. I don't remember which search gets you this, but you'll see them actually doing that. And what it is say that's myosin. You have heads all over the place, and you have actin surrounding that 3D. So it's not like you're just doing it like along this table. You're doing it all around the table. Does that make sense? Okay. So what that does, you're pulling you're pulling these actin filaments closer to the midline, and what's happening to your sarcomere? It shortens. Okay. That's the molecular level. Okay, that's a molecular level. Let's look at this. Here's a sarcomere. Here's 2. Here's 3. Here's 4. Here's 5. If I shorten this sarcomere, I'm only making it that short, right? But if I shorten all these sarcomeres, then I take something this long and make it that long. So the more sarcomeres I have in a row, the bigger my shortening effect. Okay, so if you have all of these along, let's say, a bicep brachii, that allows it to shorten in a big way. Does that make sense? Okay. Okay, this, I will tell you, is just a flow diagram with the same thing I just did with all those other pictures, if this helps you better. When we do this, those are called power strokes. Okay, those are called power strokes, and that results in literally the myosin walking along the actin, or really it's pushing the actin in this direction. Looks like it's walking along, and that's your basis for contraction. Okay. And I think that is where we will leave it. Yay!